these peptides can form. Perfect. So they can form fibrils that is like the hallmark in a brain of Alzheimer's patients. But despite that, it is believed that the fibrils are not the cause of the neurotoxicity. Instead, that the neurotoxicity comes from the oligomers that are heterogeneous. And the mechanism of this neurotoxicity of these oligomers is still poorly understood. So they are interested in studying that. So to study these A-beta oligomers is very difficult because they are unstable, they are heterogeneous, and they tend to form the fibrils first. So they have been developing a chemical model to study the A-beta oligomers. So they first stabilized these beta fragments in these macrocyclic beta harping structures, and then they covalently bind these peptides in these trimers, and then they corroborated that this structure was formed using crystallography, using size exclusion chromatography, and also by SDA space. So they established this model, and this building these chemically constrained peptides that can self-assemble could be useful to understand the molecular mechanisms that are behind these amyloid diseases. So they came to the lab because they wanted to corroborate in vivo that these trimers form these oligomers. So for that, we decided to do FLIM FRET. So you had awesome talks about FLIM already, and Lifetime with Dave Shenson, Enrico Graton, also they. So just that the lifetime is a characteristic, thing of the molecules that in our case is the time in between the absorption of a photon and the emission of that photon. And what we do in the microscope with FLIM is like we give a pulse with a pulse laser, and we excite the molecules, and then we record the photons that arrive over time, and we have this characteristic fluorescence decay curve. And you also saw the difference in between the time domain and the frequency domain. In our case, we use the frequency domain. And with our data, we did the phasor transformation that you already have seen a lot during the workshop. And some important points to highlight is that all the single exponential lifetimes will lie over the universal circle, and if the points are inside the universal circle, are multi-exponential and are a linear combination of their components. And they appear in this line showing the pure components. So for the occurrence of FRET, if we have a donor molecule that can be excited and go to the excited state, and then when decays quickly to the bottom of to the first vibrational level of the excited state, if there is an acceptor molecule that correlates in that state, the resonance, the energy resonance can happen, and the acceptor will emit fluorescence, and the donor will decay without emitting a photon. So this makes that in the phasor plot, the trajectory that what is going to happen with the donor is that it's going to be quenched, and it's going to move to shorter lifetimes. And with the acceptor, since the excitation will have a delay, since the excitation pulse, if that delay is big enough, the trajectory of the acceptor will go outside the universal circle. So some important points of the FLIM FRET is that the donor goes in this quench trajectory is curved. It's not a line, and it's not this linear combination straight that we saw before. And here we have two different possible scenarios for this quench trajectory. That in the first one is if we have a monoexponential decay that lies in the universal circle, when the molecule is being quenched, it will go towards shorter lifetimes in the phasor plot. But if we have a fluorophore that has a multi-exponential decay and the point is inside the universal circle, we can perform FLIM FRET anyway. Just the trajectory is all inside the universal circle. But in the practice, we also have autofluorescence in the cells, and we have background, and that will modify the trajectory. So in the first case, for example, if in the monoexponential case, we see that the points are going to be a linear combination in between the autofluorescence and the quenched donor. And the more you have, I'm having troubles with the pointer. The more your fluorophore is quenched, the autofluorescence is more prevalent and drags the points more towards that direction. And here we have the case when we have a multi-exponential decay. And then that also we can do quantitative measurements of this FRET and calculate the efficiency of the FRET. And I'm going to show later that with SIMFCS program, we can do these calculations straight with the program. So the first experiment that these collaborators did was they did FRET in cuvette, and they just measured intensity. They excited the donor and collected the emission of the acceptor. So in this case, one part of the trimers were bind to Psi3 molecule. They added a Psi3 to that trimer, and to some other part of those trimers, they added this Psi5 molecule. So here we can see how the emission spectrum of the Psi3 overlaps with the absorption spectrum of the Psi5. And what they excited here with, in this case, they excited with 500 nanometers, that they excite Psi3, but it doesn't excite Psi5. And in this cuvette, they were able to see that FRET occurring, and they tested in different proportions of these trimers. So then they wanted to move to the cells and see if that was happening in the biological model. So first we want to study the subcellular localization of these trimers that in these cell lines, the SH, SY5Y cells that are neuroblastoma cell line. And here we have an image with the trimers fused to Psi3, and here with Psi5, and we can see that they co-localize inside the cell. And also that, the animation went wrong, but that also we incubate the cells for 6 hours with these aggregates. And after that, we could see that some of the aggregates are not internalized. Some of them are outside the cell, but here we have a stain of the membrane, and we see some of them interacting with the membrane. So that sometimes we could see in our analysis that those aggregates that were outside, they had a different lifetime. So we could, with that, eliminate the aggregates that were outside the cells. So for our FLIM FRET experiment, we use the same donor and acceptor pair. Again, we excited with a pulse laser of 490 nanometers with a frequency of 80 megahertz. And then we detected the channel, the donor fluorescence in this channel of 550 to 600 nanometers. And then we had a second channel to detect the acceptor in the 658 to 700 nanometers, just to check that there was fluorescence over there, but all the analysis we did is just with the donor channel. So we, well, and I think I forgot to mention that we measure FLIM in our ISA instrument. And then what we have here is the first part are the controls. So we have just cells with no donor or no acceptor, just the autofluorescence of the cell. And then we have another control where we don't have donor, but we have only acceptor. And we can see that the intensity level of these two and the phasor plot position is the same in them. There is a low number of photons. That's why the phasor cloud is very broad over there. And we can see that the maximum number of photons per pixel in these conditions are 82. So it's low. So then we have conditions of where we put 100% donor and 0% acceptor, and then we increased to 50-50, and then we did 25% donor and 75% acceptor. So we had all these conditions. And you can see here in these images that we cut the pixels with a low level of photons. So all these, here you see in these images, but are in a different scale. So we cut this low fluorescence. So here we have the phasor plots corresponding to each of the positions, and maybe from far is difficult to see, but the phasor cloud started shift a little bit towards shorter lifetimes. So we characterize these phasor clouds with this SIMFCS can do this component analysis where you can extrapolate these histograms to show this phasor cloud distribution. And here the different lines are different cells for each of the conditions. So you can see that there is some variability, but all the cells behave very similar. And if we put them all together, you can see that the lifetime starts to shorter in these conditions in between the 0% acceptor or going towards the 75% acceptor. So then we use the function of SIMFCS to calculate the FRET efficiency. And here we can see the situation that we can see is the last one that I showed, where we have like a mono-exponential decay, multi-exponential decay, and all the points are inside the universal circle. And we have this quench and curve trajectory. We established the background position using the autofluorescence condition over here, this one. We put here the position, and then here we have the unquenched donor. And of course, we don't have FRET. And then while we increase the acceptor, we see that it's moving in the trajectory. And we color coded the intensity images with this here, this distribution here with this scale. And the unquenched donor is more red, and when FRET is occurring, it's moving towards this white color, and you can see pixels of the image in these both conditions with those occurring FRET. So, oops. So yeah, well, we could see that there was FRET occurring in between those trimers. So in conclusion, we could determine in a neural cell model that the peptides, they interact directly. So we validated this model of the beta oligomers so that now they can use it in mouse models to keep going and understanding the role of these beta oligomers in Alzheimer's disease.